So back in the mid-90s, when dinosaurs were still roaming the earth, Chris had this wonderful idea. What if we take a perfectly good piece of black grandma grassland and royally screw it up? And see if we can overgraze it so strongly that it crosses a threshold and never recovers. And you did this at two places. There's Stressor 1 and Stressor 2, but something happened to Stressor 1. I don't know. It got destroyed by a brontosaurus or something, and we're not still really using it. So many of you have heard of the Stressor 2 experiment. So the general question was, can we push this black grandma over a critical threshold, and then how does the black grandma respond to overgrazing in summer versus winter, and is the response mediated by shrub removal? So between what was 96 and 2000, yearly there were the grazing treatments. And you can see, looking at this aerial photo from, I think this is 2003, you can pick out the grazing treatments and the plots that were ungrazed. So there was a total of 18 plots. It was a randomized complete block design, 2 levels of shrub treatments, 3 levels of grazing, so 6 total treatment combinations with 18 total plots, 3 reps of each treatment. So the blocks were just the 6 plots at one end of the site, the middle, and the other end of the site. And it was monitored over 3 years. So Brandon got a sexy publication in a sexy French journal in 2013, actually. And so this is kind of a slam dunk. But we got answers to these questions. So first of all, for the grazing that we did, the system wasn't pushed over this threshold. And the winter grazing causes more damage than summer grazing by something like 4 percentage points. And removing the mesquite increases the black grandma cover by something like 4 percentage points. Although the paper didn't actually publish these 4 percentage points. Those came directly from the model. And there is evidence that the responses vary over time. I'll talk about more about that later. But you can see in these graphs, so this was the pre-treatment time, and then the grazing started here in these years. So a resampling in 2002, another resampling in 2009. So on the y-axis is the foliar cover of the black grandma. So you can see that in the ungrazed plots, which are in green, it increased. In the grazed plots, first it decreased, but then it started increasing again. And the trend, or the big finding from this, is that the recovery from this period, 2002 on, is at the same rate for both the ungrazed and grazed plots. So there isn't some critical threshold that has been crossed. Okay, so why am I telling you this? Well, Brandon wants to continue this study somehow. And so I guess I was interested how do we continue the study, and what adjustments, if any, should we make? And I've played around with this data a lot, so I felt like it would be fun to answer this question. So most of the monitoring for the black grandma response was done by LPI, and I know many of us in here have done LPI before. It gets really old really fast. It can be very mind-killing, especially when you have tiny sampling intervals. So the LPI method so essentially you run out a tape at whatever intervals you've decided on. You drop a pin and you record all the vegetation layers that are touching that pin. What we used in this study were 10 centimeter intervals. We had 70-meter transects. We sampled from 1 to 70 meters, which gives us a grand total of 691 points per transect, but there were 7 transects in each plot. So each of the 18 plots had 4,837 points. So on each sampling occasion, 87,066 points. And I said we only reported 3 sampling occasions, but there are actually 4. More on that later. So a total of 384,264 points. However, each point has at least 2 layers, the canopy and the soil surface. And if there's more than one canopy layer, there's even more than that. So in the entire compiled data set, we have a total of 812,443 layers that all of us poor technicians put together. Well, in the data analysis that I did, I took all of those points and aggregated. Well, I guess as your trusted statistician, first I should tell you that LPI is not actually a measure of foliar cover. It's really a measure of density or frequency. But we can derive foliar cover from it. And we found that it's actually a good way, or a very useful way, to derive foliar cover. So just simply the number of points on which black grama was observed over the total number of points that we actually sampled. So this is in any case, I guess what I wanted to say was all of those points in a single plot I aggregated to the level of that plot. So, it was like over 4,500 points I aggregated to get one number. And that one number is what I fed for each plot, fed into the repeated measures model. So this is the actual, these are the actual results from the published study. In the graphs I showed you before, we relativized everything so it had like a 0 starting value. And so my first question was, for the sake of the treatment, to detect treatment differences, did we have to sample so much? So this is what I decided to do. There's actually 4,339 points in here. Each of the plots were 70 meters square, and we had 7 transects, equally spaced about 5 meters, that we sampled. So we would start at one end, and every 10 centimeters we'd sample down. So this is the actual data, and I was wondering, what if we just did half as many points? So 20 centimeter intervals. So I subset the data and did the analysis this way. Well, what if we used 50 on down? What if we only did 49? I was just interested, what are these results going to be? Okay, so if we did half as many points, what would the results be? Well, they would have been about the same. We still have a significant shrub removal effect and significant grazing treatment effect. Okay, what if we sampled every 50 centimeters? The results would have been the same. What if we sampled every meter? The results would have been the same. What if we sampled every 5 meters? The results would have effectively been the same. You can see our p-value for the shrub removal effect is still less than 0.05, the magic number. Okay, what if we sampled every 10 meters? Well, this is no longer significant, but we still have a grazing treatment. So yeah, in any case, this pattern seems to be very strong. Okay, well, one thing that I know all of you are very mindful of this, but I think it's worth saying. All those points are really subsamples because the treatment is applied at the level of the plot. So even though we're measuring all these other points inside there, that doesn't give us more information about the treatments. All we need to be sure, or what we want to ensure, is that we've adequately measured the black grama cover inside the plots. So you might wonder, well, Darren, can't you use your fancy statistics stuff to make more use of these numbers somehow? And the answer is yes, you can use random effects to account for your subsampling, but there's no guarantee that the model is actually going to work, especially if say you use transects as your subsampling unit. If you had 0 black grama cover on a transect, that 0 poses problems or can easily pose problems in your models. You may end up getting a 0 or variance component estimates of 0, which causes which would effectively make your model over-parameterized and may affect some of your estimates. In any case, what if we, instead of having so many transects, what if we sampled fewer transects? So I did this again using just the middle transect. So first all the points and then on down all these different combinations. So if we only used one transect with the 10 centimeter intervals, we would have had the same results. If we had used one transect with 20 centimeter intervals, we would have had the same results. If we used one transect with 50, we would have had basically the same results, although this combination of removing the mesquite and no grazing is a little low. Well, what if we used 1 meter intervals on one transect, so only 70 points per plot? We're still getting significant, we're detecting a treatment difference, but some of our model-based means are a little different. And I didn't actually go low, I tried going lower than that, and the models kind of blew up because on a couple of the plots we had zeros for the black grandma cover, and the model didn't really work so well. So in any case, we could have gotten by with measuring less. Now, I don't mean this as like I don't mean to vilify anybody who made the decisions to make all of us poor techs work so hard.
That'd be me, Jared.
Even though we love to bend over and stare at the ground, it's why we get into this business. But there's actually other species that we could look at. And actually, Brandon did a patch size analysis that in the paper, I didn't actually do it, so I didn't replicate it here. But that made use of all of those extra points. So I mean, it's not that the data wasn't exactly wasted, and also you can do interesting stuff with it, like this. Okay, what's going on with the 97 sampling? If I throw that into the repeated measures model, there's some weirdness going on here. First of all, the 96 data is all down here, and then we have this huge increase. Even in some of the grazed plots, the black grandma cover increased, which seems really weird. So maybe the people who were working in 97 were like stoned or something, or who knows what's going on. We left it out of the paper. But I was interested in, first of all, I know these people, and a lot of them sampled in other years as well. So it was hard for me just to believe, it's just people are applying the LPI in a different way. Also, I was a little concerned. It never set well with me how, if we go back to Brandon's paper, how we started way down here where the control plots increased so much. Why did they increase so much? Why didn't they stay about the same? That always kind of bothered me. And I know there was a drought there in the 90s. In any case, if we go back, I lost my spot here. So I'm wondering, could this be explained by precipitation? So here is the precip from, you guys can't even see this, can you? 77 to 2010, this is the precip at Stressor 2. I also looked at Chris's notes from the beginning. Well, so the site was built in 95, but Chris's note said the continued drought postponed implementation of the main treatments. Okay, so we're talking about this time right here. Also, one thing that wasn't clear to me until I did this little exercise was that the 96, the pre-treatment data were all surveyed, there's some discrepancies in the data file. One data file says it was January and February. Another data file says it was January to April. In any case, it was basically the late winter, which would have been reflective of the cover the previous season, which was in this drought, essentially. So what I think may have happened is then the grazing treatment started happening, the shrubs were removed and stuff. But I think even in the grazed plots, the black grama would have grown more, the cover would have increased from the pre-treatment data before the cattle were actually grazing. And also, because of the sustained rains, they would have recovered from the first grazing episodes, or been able to recover from the first grazing episodes faster. What if we look at those control plots? So in the first, the foliar cover in the first surveys in the control plots was about 12%. In the second one was almost 30%. So there's a difference of about 16 percentage points. So if if those, what if those grazed plots actually also increased by that much? What would the data look like? So I tried this little exercise where I added 16.06 to the 96 plot cover estimates. And this is the result. And this may actually be more like what actually happened. That's what I'm guessing. In any case, I'm not, this doesn't undermine any of the efforts that have been done so far, or Brandon's paper or anything. But I think it's very instructive about how, with regard to how we continue the study. So my recommendations, first of all, our sample size is really already fixed. So if we have a new question, first of all, is our n of 3, or considering the block, or n of 1, really sufficient to answer it? I don't know what, Brandon, what your new question is going to be. But maybe we should plan more explicitly for these periodic fluctuations, like Don was talking about, this compressed growing season. The black grama can respond, faster than the shrubs, for example. So if we, but on the other hand, our pre-treatment data is locked into that late winter period. So we're, maybe there's nothing we can really do about it. Avoid the LPI sampling while the grazing treatments are taking place. So that 97 sampling was right in the middle of the grazing treatments. And maybe it would be worth, so the grazing treatments lasted for 4 years. Maybe it's not worth doing the LPI sampling during that time. So sample the same transects in the first year. There are actually 14 labeled transects in each plot. In the first year, 7 of them were randomly selected. I wouldn't advocate that. I think sampling the same ones would be better. And perhaps consider increasing the LPI intervals or reducing the number of transects. And if Brandon, if you're specifically interested in some patch dynamics, I don't know, maybe LPI isn't necessarily the best way to get at them. Also, I just made this like 5 minutes before I came out here. I was thinking, well, let's look at the basal cover. So these are the basal hits. Now we have a mess here. So LPI, at least the way it's been, I think the foliar cover estimates may be fine. But relying on LPI for estimating basal cover is probably not the best thing to do, at least given what the data are saying here. So thank you for paying attention.